everyone. My name is Lindsay Shuck. Thank you for joining today's Family Office Network's webinar series featuring Akamara. Presenting for us today is Susan Graff, CEO. Susan is an accomplished executive in global biopharma with 25 years of broad industry experience in both large pharma and small biotech. Susan has therapeutic expertise in development and commercialization of biopharmaceuticals, new product forecasting, financial valuation, due diligence, and deal making. Susan earned an MBA from the Stern School of Business at New York University and a Bachelor of Pharmacy degree from Purdue University. At the end of today's presentation, we will have time for Q&A. Please use your questions chat option on your screen to send over any questions you have so we can answer them accordingly.
Thanks, Lindsay. Good afternoon, everyone. It's my great pleasure to introduce you to Akamara Therapeutics, the company that will be known for reimagining immunotherapy. We're going to do that first in cancer by really providing inventive B-cell therapeutics that change the way people live. The team, you heard a little bit about my background, but the team behind this spans the gamut from early science with our founders, Shiladitya Sengupta and Dr. Monadeepa Roy, to commercial experience with myself and Sergio Santillana. You heard my background, but Sergio Santillana, I just want to highlight, he has a number of different oncology approvals to his name, and he is really our lead for the phase 1 clinical development program you're going to hear about today for our lead program, AT1965. Chandrasekhar Natarajan, another very experienced industry veteran, he's been leading our translational medicine efforts and really driving our biomarker program that we're also going to study in our phase 1 study. And Jim McArdle, again, experienced industry contributor here who's running our CMC efforts and responsible for producing the phase 1 material that we'll use in our clinical study. We supplement the internal team with this esteemed panel of scientific advisors. We have Dr. Bruce Zetter, here local in the Boston area where I sit at Dana-Farber, who helps drive our cancer biology efforts. We have Dr. Ricardo Dalla Favera down in New York at Columbia, who is a renowned B-cell expert who we tap into when we're interrogating this novel biologic approach that we're pursuing. We have Dr. Shireen Loy, who is a translational expert. Particularly, a lot of her work has been around T infiltrating lymphocytes, and she's also very knowledgeable in B infiltrating lymphocytes. She's based in Australia at one of their large cancer centers, Peter Mac. And then Aditya Bardia, here locally again at Mass General, very experienced oncology principal investigator. So there's a wealth of experience from both the internal team and our scientific advisors that have been lent to advancing this program. In terms of where our lead program, AT1965, is, this is a very exciting novel B-cell activator for cancer immunotherapy. Earlier this year, we had a pre-IND interaction with FDA and received very positive feedback from FDA, really clearing a path to a 2020 IND. We plan to follow that up with a first-in-human study in solid tumor patients. Again, this is a cancer immunotherapy approach, so we intend to dose a number of different solid tumors within that study, which I'll tell you more about when we get to that part of the presentation. Accel, the large tech venture group, was leading this company's Series A and continues to be a significant shareholder in the company. Back in 2018, the first tranche of the Series A was raised. That was $9 million. Second tranche was taken down in 2019, that was about $6.5 million. And we are currently looking to extend our Series A on the same terms as that second tranche, so a $29 million pre-money valuation, and really looking to raise approximately $15 million US to advance AT1965 into the clinic. Before we jump more to our program, I want to frame for you the immuno-oncology space because it's very crowded and it's rapidly evolving. What you see here on this slide is a schematic on the left that was actually taken from a Nature publication that was looking over time as to what are the most common cancer immunotherapy approaches. And you can clearly see that T cells and T cell approaches absolutely dominate the cancer immunotherapy field. There's good reason for this. There are approved products here. The checkpoints, the PD-1, PD-L1 fall into this category, as does CTLA-4. CAR T also falls into that T cell category. Very different cell therapy modality versus the checkpoint antibodies, but nonetheless, that has been where the bulk of development has happened. But now, as we learn more about the human immune system and what's needed to really activate the immune system against cancer, there has been more and more exploration on other immune cell types far beyond T cells. And you see that listed here as well. You see, if you start at kind of the far left, if we just focus on 2020, the macrophage bubble is fairly large and growing. We have the tumor infiltrating lymphocyte there at the bottom in pink, TIL, that's also growing. We have natural killer cell approaches, NK in kind of the yellow bubble, top middle, that's growing, neutrophil approaches. And if you look kind of right dead center, little tiny bubble under 2020 called B cells with 18 programs listed there. It is a newer area that's becoming more active, but yet not fully explored. So we have a really unique opportunity to lead into the clinic here. And what I can tell you with AT1965 is, to the best of my knowledge, we are the only B cell activation approach that's being applied in solid tumors. These are large tumors like breast cancer and lung cancer and colon cancer. Most of the B cell approaches that you would see listed here under this bubble are B cell approaches that focus on hematologic malignancies like lymphoma and multiple myeloma. And their goal is to actually destroy the B cell, because the B cell or the plasma cell is actually cancerous. That's absolutely not what we're doing. We're taking a very different approach to try to activate B cells against solid tumors, much like what checkpoints did with T cells in solid tumors when they first came really into this marketplace. There's very good reason why we need to move outside of T cells. If you look at the chart here, what you see on this green line is really the proportion of cancer patients across all different tumor types that are able to achieve what's known as the long survival tail. That means, out past a 5-year survival rate, which you see shown here. It's really still only roughly 20% of those patients who receive the currently approved cancer immunotherapies that are able to achieve that long survival tail. And there is a lot of interest in exploring these other immune cell types, because we don't know if other T cell targets beyond PD-1, PD-L1, CTLA-4, we don't know if those T cell targets will be able to move the needle here, and hence the need to go after different immune cell types. And as you clearly can tell, we've chosen the B cells and to go there. And what is really exciting about that is we are not only leading into the clinic with our program, but we're really riding the scientific wave around B cells and their role in solid tumors. What you see here are kind of some snippets from a Nature series published in January of 2020. You can clearly see on the left there the kind of headline, B cells to the forefront of immunotherapy. This was a review looking across a number of different solid tumors, melanoma, sarcoma, and talking about the role that B cell activation and infiltration plays in relation to outcomes, and including survival in those tumors. The Nature series is just one piece of evidence of this growing scientific wave. There's a number of other publications, as well as our own work, that really cements the B cell role and its possibility for a therapeutic approach in these human solid tumors. We know that B cells are antigen presenting cells that can produce tumor-specific antibodies. We also know that B cells are unique in that they can really elicit a durable memory response to an antigen, such as a tumor antigen. We see when you have B cells and tertiary lymphoid structure, which is kind of a B and a T cell cluster, infiltrate into the tumor, that you are able to predict a response to checkpoints. Because when you see those structures in the tumor, you will likely have a positive response to a checkpoint inhibitor. We also know that B cells can be quarterbacks in our immune system, and I'll show you more about that on a coming slide. But when I say quarterback, they can coordinate other cell types and hence promote also the release of cytokines and things that are driving cytotoxic T cells to kill tumors. And we also are learning more and more about how B cells may play a direct role in shaping tumor responses. They may have ADCC properties. They also likely elicit phagocytosis handled by other cells in the immune system. This is the slide I meant when I said B cells can be quarterbacks. I'm not going to walk you through every component on this slide, but I think it's important to ground you here because a B cell approach is very different than some of the other immune cell types. If we start on the far left, you can just see their B cells with all those arrows coming off of it. Again, this just shows the number of different interfaces that B cells can drive and kind of quarterback in the immune system, including key things like complement activation, antigen recruitment, antibody production. When we look in the middle of this slide, what we see is a maturation process for a B cell. And this is actually very important because we know a lot about this process from viral infections. Originally, when you're infected, you have a population of naive B cells that ultimately identify a foreign pathogen, like a virus, or in our case, when we're thinking about cancer, a tumor. And you move through this process of B cell activation, ultimately antibody secretion, and ultimately the creation of memory B cells, which will then produce antibody again if they're ever insulted again with that foreign pathogen. We see a similar response to what's shown in this center slide when we treat cancer with our compound AT1965, again indicating that kind of B cells are the quarterback in the immune response we're inducing. On the far right side of this slide, again, not going to go through every piece because this is a really growing area of research, not only for us, but you can clearly see for others because this is from a recent publication earlier in 2020 as well. But the understanding of how these different immune cell types, B cells included, can actually attack and kill a tumor continues to evolve. We know that that's absolutely possible with things like cytotoxic T cells. What we really want to understand more with regard to our compound is exactly how are B cells doing that beyond the potential antibody secretion and recruitment of other cell types like macrophages and perhaps cytotoxic T cells. I mentioned that we induce a pattern in cancer similar to the immune response to a viral infection. Very specifically what I mean by that, and you can look back on this presentation and track this slide with that center panel. We see increased IgM levels early in treatment of cancer with our compound. We then see a very strong interferon response, which again would be expected, and see things like the interferon regulatory factors go up and interferon stimulated genes produced. And in our case, one of those key interferon stimulated genes is a protein called IFIT1. We see elevated dendritic cells and activated MHC2 positive macrophages and monocytes migrate into the tumors when we treat with our compound. And we see a decrease in B-regs, which we want to see. And we see an increase in memory B cells. And all of these things, again, indicate that B cells are the coordinator for the immune response that we're seeing. So we're very excited that we've been able to kind of pull our program outside of this T cell fray and differentiate and lead into the clinic here with a B cell activating approach in solid tumors. In terms of our specific molecular target and mechanism, we can only share that under CDA because it is an absolute novel oncology target. What I can tell you is that we are a small molecule. We have US composition of matter patent valid through 2033 without extension, as well as filings for global patent protection through Europe and Asia and all key regions of the world. I'm now going to take you through all of our preclinical animal in vivo data that further shows our excitement around this compound in a variety of solid tumor settings. What you're looking at here are 3 different solid tumor models. Far left, noted by TNBC, is triple-negative breast cancer. It's the 4T1 tumor model, and all of these are syngeneic mouse models. 18-1965 is shown in the blue line, and you can clearly see relative to control in triple-negative breast, very nice tumor reduction with 18-1965. That's unusual. We typically see more tumor growth inhibition in a model like this because this is a very difficult to treat solid tumor. So we're very excited about that data. When we look at melanoma in the middle, you see your more classic tumor growth inhibition with 18-1965 in the blue line. And Lewis lung carcinoma, again, nice tumor growth inhibition. Taken together, this gives us confidence that the molecule has activity across a number of different solid tumor types. When we start to really interrogate the immune memory that we're creating by treating with 18-1965, we look at this experiment. The top row of mice here is our experimental group, and then the next 2 rows are our control groups. We've used the 4T1 triple negative cell line here. We've implanted the mice with tumors, treated with 18-1965, and we see those tumors regress, just like we did on the previous slide in that triple negative breast model. We then waited 25 days and re-challenged that experimental group of mice with 4T1 tumors. And what you see are the results here in that line plot on the top right. You see that initial tumor growth and then the regression and then no further growth upon re-challenge, so out to day 70. So we believe that that is because immune memory to the tumor we implanted was created in the first treatment experience here. That's in stark contrast to the control groups, which are noted in terms of tumor growth on the bottom right line plot here. The reason we had 2 controls, they're both non-tumor bearing controls. We treated one with our compound, 18-1965. We treated the other with saline, and then challenged both of those controls with 4T1 tumors at the same time point we challenged the experimental group. And I think it's just important to note that the tumor context and the sequence of treating here is important, meaning there's no prophylactic protection provided by 18-1965. It has to be an experience where the immune system has recognized the tumor and 18-1965 has amplified, if you will, that immune response. Here we're looking at a variety of different immune contexts in the 4T1 tumor model. The reason we did this again is we wanted to see what would happen if there was not an intact functioning immune system. So on the far left is our immunocompetent wild type model, where 18-1965 is in the purple. And you can again see a nice tumor growth inhibition relative to control. In the middle panel, when we knock out the entire immune system and use SCID mice here, so no B cells or T cells, you see 18-1965 loses all of its activity. So it's really superimposable there with control. This is important because a functioning immune system is important for the compound to have meaningful activity. We do not see direct cytotoxic activity like you would see with a chemotherapy, for example, without a functioning immune system. And when we move to the far right panel in the B cell knockout, this is a JH model, you see again the compound loses the vast majority of its activity, again indicating that B cells are the quarterbacks here. that are instigating this entire immune response we see that results in tumor growth inhibition and tumor regression in some models. We followed that set of experiments up with yet one other experiment looking at the B cells and their ability to confer immune memory. We call this our adoptive B cell transfer experiment. And the design here is an experimental group of mice implanted with 4 T1 tumors, treated with AT1965, and then when those tumors had regressed, we harvested the spleens of those animals, isolated what we call primed B cells, primed because they've seen tumor in our compound, and we then injected those primed B cells into a new experimental group of non-tumor bearing mice. And then we challenged those mice with tumor. You can see the results in the line plot here on the right. The light purple line is the bottom row of mice here that you see, where they had some initial tumor growth, and then the primed B cells we gave them caused tumor growth regression. That's in stark contrast to a saline control and a naive B cell control, which you can see in the blue and the green lines here. Again, the B cells have to be able to have been primed by the tumor and our compound previously to have any anti-tumor effects here. The other thing that we're very excited about from our preclinical work as we head toward the clinic is the fact that we have activity even in a scenario where T cells may be fatigued. This is, analogous to if a patient has received previous checkpoint therapy, perhaps their T cells are a bit fatigued. Not a perfect correlation because there is no perfect animal correlation there, but what we did was looked at what happened when we deplete in a 4T1 model T cells using anti-CD4 and anti-CD8 antibody. And you can see that here, that AT1965 does not lose significant activity in the context where that compound is given and the T cells have been depleted. So you can see that in the maroon line, and then AT1965 alone in the blue line. This was exciting for us just because T cells are clearly important in cancer immunotherapy. I'm not saying they aren't, but our B cell activation approach is an interesting way to potentially elicit anti-tumor activity that is somewhat not fully dependent on T cells, which was very exciting for us as we think about going to the clinic. In terms of where we are with the program, we have pre-IND meeting feedback from FDA in hand, and that was very positive. Most of their comments really were around our phase 1 program. We've completed our rat and dog GLP toxicology studies. We've produced our GMP drug substance and are teeing up to produce our GMP drug product, which will be the material used in our first-in-human study. We've drafted all of our IND modules, and we've selected our CRO to run our first-in-human study. So again, targeting a 2020 IND followed by a first-in-human study start. Again, the fundraise we're looking to do here really is meant to propel us from where we are today into that first-in-human study and through that first-in-human study. The plan we've outlined for phase 1 is a classic dose escalation. Again, it's a first-in-human, so we have to first determine the safety of the compound, followed by dose expansion, which would be more specific tumor cohorts. I'll share with you in a minute the tumor types we're going to be enrolling, but in short here, we're envisioning 100 patients will give us a meaningful clinical read. It's going to be about 40 patients through our dose escalation group, and that will take us through what we believe are efficacious doses to a maximum tolerated dose. That'll take us about a year once we start the study. And then we're following that up with roughly 2 to 3 expansion cohorts. And we'll help select those cohorts based on what we see in the dose escalation phase. But we expect, that it'll be roughly 20 patients, slightly larger if we only go with 2 in each of the expansion cohorts here. That'll give us another set of 60 to 70 patients that will really help clarify whether there's not only a safe compound here, but a compound with single agent clinical activity. That'll take about another 12 to 16 months here. So we're looking at roughly a study timeline to get some meaningful data from the time we start of about 24 months. So there's also a near-term turn here in terms of this not likely being a 5-year endeavor before there's an opportunity for a next value inflection. Now, in terms of the study tumor types that we plan to enroll, you see them listed here. We've actually broken them into 2 columns by design, but what I will say is that all of these tumors are known in the literature to have a positive role on outcomes like survival when we see B cell activation and infiltration. So that was one of the key lenses we used here. We wanted to select tumor types where we know from literature that B cell activation, it matters. On the left are some of those large tumor types like triple negative breast, non-small cell lung, malignant melanoma, and colorectal carcinoma. You've seen activity of AT1965 in 3 of those models. I can tell you in colorectal, it's also nice tumor growth inhibition. So we have demonstrated activity there. We've supplemented that tumor population with those on the right that we've selected from literature. And these on the right, in some cases, are smaller, underserved tumors, or they have fewer treatment options. And the regulatory path could also be faster or different if we see a signal, for example, in cholangiocarcinoma, which is really kind of a bile duct cancer. There are not a lot of treatment options for that. And that regulatory path, if we saw clinical activity there, would likely be very different than something in non-small cell lung cancer. So we've given ourselves some strategic advantage here as well, depending on where we see the clinical activity. I will just also point you to the literature here before I close, that the statements I've been making about correlation with outcomes like survival, they are absolutely supported in a variety of different literature. What I've shown here is a Kaplan-Meier from Clinical Cancer Research. It's an estrogen receptive negative breast cancer population. And they're actually looking at a mark surrogate for B cell activation called IGKC. It's uniquely expressed on activated B cells. And you can clearly see that patients that had high IGKC expression here had better survival than those with low IGKC expression. So when we think about biomarkers in our study, IGKC is one of the marks that we'll be tracking to not only look at things like partial and complete regression with regard to the tumors, but also when are we hitting a threshold of B cell activation that we can measure. I will just also share a case study here that shows for cancer immunotherapy, clinical data is very important and clearly creates value. A mouse immune system is not like a human immune system, and we always have to have the confidence that the immunotherapy approaches are meaningful in patients. And that's why getting through this next study with our compound AT1965 is so important. But here you'll see, just very briefly, there was an acquisition in March of 2020. Gilead came and paid nearly $5 billion for a company called 47. Now, 47 had only really kind of started 5 years earlier, 2015. They are going after an anti-CD47 antibody program. And anti-CD47 is not a B cell target, but it is a novel non-T cell target that the company was able to deliver positive clinical data on. And that led to that confidence that this is a validated cancer immunotherapy approach. That's what we're trying to get to in terms of being able to generate some meaningful positive clinical data with this next round of funding. So hopefully this was a good overview for you, not only with our program AT1965 to show you that we're kind of near clinic ready, that we have a very differentiated approach versus the other players in the cancer immunotherapy field. We are a simple, small molecule, not complex cell therapy. We haven't talked a lot about that, but our manufacturing will be much more straightforward. We have the promise of anti-tumor activity across a range of different solid tumors. And with this positive FDA feedback in hand, confidence that we can move through an IND this year. The program's been steered by a wide range of experienced industry professionals to this point, and for you, our expansion of our Series A is an opportunity to really invest early at an attractive valuation with all of these key milestones in front of the company. And with that, I will close and take any questions.
Susan, thank you so very much. As we would like to open the Q&A section of the webinar today, again, to all of our attendees, please use your questions chat option on your screen to submit those questions over to Susan. Susan, let's kind of just jump right into it. First question here, are there any other companies pursuing the B-cell activating therapies right now in cancer?
So we've identified some very early kind of discovery work, but companies know that has actually been identified at academic institutions. We do believe that we are light years ahead leading into the clinic here with B-cell activation in solid tumors. The other B-cell approaches, as you saw kind of on that bubble slide, those really are, to my knowledge, targeting heme malignancies, where you're depleting B cells. They're not going after solid tumors, nor would that be appropriate because they're depleting malignant cells. They're not able to activate B cells.
Okay, perfect. Thank you so much. Let's see, moving on to the next question. How long do you expect to be in the clinic before you start seeing clinical activity?
So we've defined what our dose steps will be for that dose escalation phase. I can tell you that we will need to step through probably 3 dose levels before we hit what's a potential human efficacious dose. That will take a couple months, but you saw that dose escalation is meant to be roughly a year. So I can't pinpoint it for you because we have to ensure patient safety, but it will be not at the end of our dose escalation period. We plan to dose well past what would be a potential therapeutic dose to try to reach a maximum tolerated dose. So something less than 12 months.
Great, thank you. Let's see, next question. What are the advantages of a B cell activation to cancer versus the T cell approaches?
So it needs to be studied in humans. We firmly believe that, and that's been our path, is fast to the clinic so we can generate the human data. Obviously, the T cell approaches with checkpoints are well known. We do think if you go back to the T cell depletion data I showed with our compound, we do believe that this offers a very different way to stimulate the immune system against the tumor that is really, I don't want to say fully independent because there's so much crosstalk in an immune system. T cells are absolutely important, but what we learned from that is that the B cells can coordinate an appropriate anti-tumor immune response. And we do think that that could hold a lot of promise as we think about patients who've previously been treated with checkpoints. So think about patients who've already been diagnosed with lung cancer. They've already received a platinum chemotherapy and a checkpoint because that's very much standard of care now in lung cancer. What are those next treatment options for those patients? There aren't many. We think that's very much where, at least initially, something like a B-cell activation approach could come into play.
Okay, wonderful, thank you. Next question, can you elaborate a little bit more on the ask today, the raise that you're looking for?
Again, we're looking to actually expand our Series A. To date, the Series A has only taken in $15.5 million. We're looking to expand that by about another $15 million, 1.5, so be just over a 30 million Series A. And that will really get us into the clinic and largely through the study that you just saw. We are looking for the same terms as the second tranche of the Series A. So the pre-money on that was roughly $29 million.
Perfect, thank you. That seems to be the last question that pulled through so far. So we can take a few minutes to see if there's anything else. Susan, would you like to add anything while we kind of pause here for a moment?
No, I would just close by saying that if you're interested in cancer immunotherapy, I really do believe that Akamara, an investment here, offers a way to diversify your cancer immunotherapy approaches and offers huge promise for a paradigm changing approach in the field that will be realized in the very near term.
Wonderful. Thank you, Susan. Thank you so much for joining us today. And to all of our attendees, thank you so much as well. Again, this is Lindsay with Family Office Networks, and I will be following up with everyone individually so I can get you in direct contact with Susan if there are any additional questions. We hope you all have a fabulous day, and we look forward to speaking with you soon. Thank you.